All right, so we are live, everyone. My name is Jesse, and I am with Exploring by the Seat of Your Pants. If you're joining us for the first time, and I know we do have a few people joining us for the first time, we are all about bringing conservation, adventure, and science into classrooms around the world. Thank you so much to all our viewers for tuning in amidst the summer. Again, we typically operate September through June. We've been thrilled to present a few really exciting presentations as the summer rolls on, so thanks so much for joining. Duke Lemur Center has been one of our biggest partners over the last few months. We have highlighted so many amazing stories about lemurs, about Madagascar, and more. But I'm particularly excited because we have this really cool 4-part series going on right now with Dr. Lydia Greene and her partner, Dr. Marina Blanco. They are going to be talking about all sorts of the elements of lemur science, Lemur Science 101. Last week, we did the why. Why lemurs? Why Madagascar? Why is this such an exciting and important place? Today, we're going to dive in on the how. So how do you go about going through the rainforest to track a mouse lemur in the middle of the night? Or how do you go about picking poo apart to find seeds? Why on earth are you doing this? How do you go about it? How do you get there in the first place? So I'm going to turn it over to the relentlessly enthusiastic Dr. Lydia Greene. She's going to blow our minds. Thank you so much for joining us today, Lydia. And I can't wait myself. Take us away.
All right, thanks for having me. And I think relentlessly enthusiastic is something I'm going to be adding to my cards. So yeah, I'm just going to go ahead and pop on screen share automatically and pop up the slideshow. Jesse, does that look good to you?
Looks perfect. Let's do it.
All right, so thanks everyone for joining. I'm really excited to be here for the second part of our series, Lemur Science. Today we're gonna round out our first introductory seminar of Lemur Science 101, and then next week we're gonna progress to Lemur Science 201, so we're gonna graduate to a much higher level. Right, and so like Jesse said, last week we talked about the why lemurs, why Madagascar, why are scientists endlessly fascinated by the system, and today we're gonna jump into the how. How is it that scientists learn all this stuff about lemurs? How do we know what's going on? And so that's really the main theme of today. How do we know this stuff? How do we as lemur scientists learn this stuff? And the most important part is that it begins by having a question. Curiosity is the bedrock of science, be it lemur science, be it space science, be it marine science, be it any kind of science. You have to just start with being curious about something. And for lemurs, this curiosity could take a million different forms. There could be a million questions you could ask about lemurs. And so I popped up a few, just a handful on the screen, so you could get an idea of how many things we could go about learning in lemurs. Maybe you're curious about what that specific lemur is doing. Maybe you want to know, how do lemurs communicate with each other? Why are they so colorful? Are they smart? Why are there so many lemurs? Why are they fluffy? What happened to the giant lemurs? What food do lemurs eat? Do snakes eat lemurs? There are so many questions that we could go on asking in lemurs. And some of these questions we've been asking them for hundreds of years. And maybe some questions you might have never been asked before. And that could be something you could spend your entire life pursuing, which is sort of what happened to me. And so today what we're going to do is we're going to take just a couple questions and trace them through from question all the way to collecting the data, generating the evidence that we would need to answer that question, and then showing how we can use this method to really figure things out about lemur science. And so we're going to start with what might seem like a really simple question, which is, what do brown lemurs eat in the rainforest? And you're going to find out that it might not be that simple to solve this question after all. And just a semantic point first. When I talk about brown lemurs, this gets really confusing because it doesn't refer to the color of the animal. So there's actually 12 species of lemur that we refer to as the brown lemurs. Some of them are black, some of them are like honey golden color, some of them are more red, but they're all called brown lemur. So just when I use that term brown lemur, I'm referring to this group of lemurs, not the color of them. And in particular, we're going to ask this question of, what do brown lemurs eat in the rainforest of this lemur species, the red-bellied lemur. This is a female red-bellied lemur. And also of this species, the white-fronted brown lemur, so brown back but white head. And these guys happen to live in the same rainforest as each other in the Northeast. And so we're going to study them at the same time. And we're really curious about what they're eating. How is it these animals are able to extract energy from their environment to go about and live their lives? Obviously, they can't go to the grocery store the same way that we can. And so there's a couple of different ways we could do this, or we call methods. And so one obvious method would be to go into the forest and observe these animals eating. If we can figure out what they're stuffing in their face and what they're chewing and swallowing, we can figure out what they're ingesting, and we can use that as a way to reconstruct what their diets are. And so that seems like a pretty obvious way to go about doing things, and we're going to do that today. But there may be some less obvious ways you could think of, like examining their poop, because what goes in must come out. And so we can actually use feces as a way to reconstruct what lemurs were eating to begin with. And it could be that by studying poop, we can get answers that we couldn't get to this question just by watching lemurs alone. And so you'll see that as well today. Okay, so now that we know which lemurs we're interested in studying, we have our question, we know what sort of our methods are going to be, we get to go to the forest and collect data. And so in particular, these brown lemurs live in the rainforest, and they live specifically in the northeast rainforest. And so we're going to travel to this site called Marajeji National Park, which is this huge, pristine, beautiful rainforest ecosystem up north that I've had the real privilege to get into. And if we look at Marajeji from sort of a bird's eye view of the map, you can see it's this enormous, massive ecosystem that's there. It's very mountainous as well, so we're going to spend some time hiking. And so what we do is we travel to the coastal city of Sambava that's along this yellow road all the way on the coast. And so you can travel to Sambava, and there you can stock up on supplies like food. And we take a 3-hour drive along the countryside, along this really nice paved road to Marajeji. And along the way, you get these beautiful vistas, and you start getting closer and closer and closer to Marajeji, which is this mountain chain. And eventually, after about 3 hours into the drive, you arrive in this village called Mandena. And Mandena, you can now see just how close we're getting to Marajeji, which is this mountain in the background. And Mandena is this community that lives in the shadow of this mountain. It's the closest village that you can get to into the sort of tourist or main entrance to Marajeji National Park. And it's just an amazing place to consider living, being in the shadow of this mountain. And in Mandena is also where we usually meet the rest of our team. So for this particular mission, we might be teaming up with a bunch of guides. So Des Kelly is always one of our senior guides. He knows this rainforest like the back of his hand. We might also work with another guide like Yokono, who's really enthusiastic and eager. January, who is an expert tracker. He could find lemurs anywhere. And Rabe Smoke, who's our cook, who makes excellent dishes for us to eat, so we're not cooking and we can focus on our work. And so we meet our team in Mandena, and then as a team, along with porters who help carry our stuff, we set off to Marajeji. And even though Mandena is really close to Marajeji, it's still about an hour hike to get to the park entrance from this village. And this is actually a really nice hike. The only downside is you're like out in the sun, so I tend to get very burnt on this hike, but it's this beautiful hike through agricultural fields. You get to see how the community practices rice farming or vanilla farming or other types of agriculture. You can see their zebu or their aumbi, those are the cows that are out in the rice fields. You get to go on these paths along the river and see lots of fruit trees. And this is the main river that is the source of water for this community that you get to sort of travel across as well. And it's really kind of an exciting hike. And then you arrive an hour later at the entrance to Marajeji National Park. And usually by this time, I'm actually pretty tired already. And so you arrive at the entrance, and then you realize that like, we're gonna hike up to Camp 2. And so Marajeji is weird among a lot of the parks in Madagascar, and that most of the parks you sleep outside the park in a hotel or at a campsite, and then you go into the park, you take like circuits, maybe a 4-mile circuit, and you see the lemurs during that circuit, but then you come back out and sleep outside the park. But because Marajeji is so big, we actually sleep inside the park, and there's 3 camps that you can sleep at, and you reserve ahead of time where you're going to sleep. So there's Camp 1, Camp 2, and Camp 3, and we usually push to get straight to Camp 2 because that's really the heart of the park and the heart of where a lot of the lemurs are. And so Camp 2 is now a further 4-mile hike in, which doesn't sound that bad until you realize that you're also going to go half a mile up. And most of that half mile up is in the last mile. And you end up like finding these ropes courses that you're like hauling yourself up. And I usually fall down a lot and everyone laughs at me because it's funny. And it's actually really hard to get up to Camp 2. But the reward is totally worth it because the vistas that you get at Camp 2 are stunning. This is pristine rainforest at its best. It is mountainous. There are birds chirping. There are insects buzzing. There's also a waterfall at Camp 2 that you can just sit on these rocks and hang out and stare into this incredible view. And you get there and you're usually pretty tired. So Marina and I will tend to crash on the rocks. And as the sun starts to set, you realize that it's actually quite cold now because you're pretty high up. And so you usually find Des hanging out by the cook stoves to stay warm. Rabe Smoke usually cooks a great meal, something beans and rice, like a nice stew. There's tea, and then you settle down to sleep for your first night. And we usually go to bed right when the sun sets at like 7 or 8 o'clock, and you're just wiped by that point anyway. And so you wake up in the morning, and January's already gone off to find all the lemurs and calls back their location, and you go out and find the lemurs, and like you have this realization of, oh my God, we found the lemurs. This is amazing. This is awesome. And then it's like, now what? Because for many of us, when we travel to a place like Madagascar as tourists, the ultimate goal is to see the lemurs. And so you spend all of this effort just trying to find the animals. But the first time you see them, then you can check your box, you found the lemurs. And for us researchers, it turns out that this is now just the beginning of our journey. So flying to Madagascar, getting to Sambava, picking up your supplies, driving to Mondana, meeting your team, hiking to the entrance, hiking up, sleeping overnight, finding the lemurs. That is what we have to do to begin our workday. And so now we've got the lemurs, now what? Well, of course, we're going to do behavioral observation. We're going to record their behavior. And so really what that looks like is like these scratchy handwritten notes in my notebook. We usually take our notes by hand. That way we don't have to keep batteries charged or worrying about devices failing. So we still use a lot of handwritten notes. And these are some of my more scraggly notes that I pulled out for this photo. And so it might seem obvious now we're going to collect data, but there's actually a lot of decisions we have to make about what we're going to record. So what are we actually going to write down in these scraggly handwritten notebooks? And so we could write things down like what they're eating. We might record if the lemurs are eating fruits or leaves or insects, but we might want more information than that. Maybe we want to record the age, the size, or the color of those items. Is it unripe fruit? Is it a young leaf? Is it an old leaf? Is it a red fruit versus a black fruit? Maybe all of those things are important for us building this knowledge about what these lemurs are eating. And we might want to identify it. Is it a maple tree versus a coconut tree? And obviously those are some fruits that I'm familiar with, but this is where your guides in Madagascar can really come in handy, because a lot of what grows in Madagascar doesn't necessarily grow anywhere else. And your guides are really going to be the ones that have the knowledge to tell you what a lot of these plants are. You might be interested in other pieces of information. How many items did that lemur eat? Did it eat 3 leaves in a minute, or did it eat 30 leaves in a minute? And maybe that's really important for the kinds of energy they're getting from their environment. And you might also be curious about how long they spend eating. Are they just eating that for 10 seconds? Or are they eating it for 10 minutes? And then you might also be curious to think about who you're going to be studying, who's doing the eating. How many lemurs are you actually going to follow? Are you going to watch just one lemur? Are you going to watch 10 lemurs? Are you going to watch 100 lemurs? Are you going to watch all lemurs from one social group? Or are you going to watch many different social groups to try to figure out how these different groups are doing things? Are you going to study both males and females, or just one or the other? And are you going to prioritize animals of a particular age, say young animals or adults or old animals? Or maybe you're really interested in juveniles and what juveniles are doing. And maybe you also want to make sure you can follow the same animal consistently across days. And so you need to be able to identify and find that animal over and over and over again. And so this is where something that we use called collars become really, really important. And so I've got this video of Wig chewing on collard greens. It's sort of a lemur video to put on while I talk about some of this more boring stuff. But we tend to use these collars a lot on these lemurs for a couple of reasons. And you've probably seen our lemurs at the Duke Lemur Center wearing these collars as well. And so collars serve 2 main functions, which is that one, by putting this red collar on Wig and not putting a red collar on anyone else, I know exactly which animal in the social group Wig is. I can always identify her because she's got this collar on. And so it makes it really easy to follow the same animal over and over and over again. And this might not seem that hard if you've only got, say, 2 animals in your social group, but if you have a social group of like 15 animals, it can be really hard to tell who's who. And then the other purpose served by these collars, or you can sort of see on the right-hand side of her collar, well, our right-hand side, is there's like a little like yellow bit on her collar. And this is actually a radio transmitter. So as she turns her head, you'll see it again. There we go. And this radio transmitter is also really important Oh, sorry, wait, go back. Because this radio transmitter emits a unique frequency. So each caller that we order is going to have a unique frequency associated with it. And so this caller is sending out that frequency into the world. And then we can use what's called radio telemetry to track and identify that signal. And so we, from many, many, many miles away, can use our antenna and our receiver to pick up that beep. And that's going to tell us which direction we can find the animal, and it's also going to tell us which animal it is. So these collars are a really safe and really critical way for us to be able to find the same lemurs over and over and over again, and also to find the lemurs and not waste time like sort of aimlessly looking through the forest to see if we can figure out where they are. We know exactly where they are because we can track them. All right, so once you figure out who you're going to follow, we have a couple of more details to get through, which is when are you going to do this study? Are you going to study animals in different seasons, like the rainy season and the dry season? Because maybe they eat different things in different seasons. What time of day are you going to be studying them, in the morning or in the evening? Because obviously what I eat for breakfast and what I eat for dinner are two very different things. Usually pancakes features heavily on one of those. And then you also might want to figure out how long you want to watch the animal. Are you going to, how long is enough time? Is an hour enough time? Is 10 hours enough time? Some of these animals are also awake at night. So do you need 24 hours to watch them? And if that's the case, when are you going to sleep? So all of these are decisions you have to make when you're designing your study. And then there's one other thing that's called the how do you watch. And this gets into the meat of how scientists collect data. And there's two major methods we used called the focal method and the scan method. And the focal method is exactly what you're thinking. We pick one lemur, like Wig. She's got that collar on. We know we can find her. We pick one lemur and we follow that lemur for a period of time, say 10 hours, and we record everything that Wig eats. We intensely focus on just Wig, and we have this great record of everything Wig did during that day. But the problem with the focal method is that it doesn't tell us what anybody else besides Wig did. So we only have a record of what one lemur was doing. And maybe that's not that useful for the particular question we have. So then we can use this other method called the scan method. And the scan method is where we don't focus on one animal. We focus on a bunch of animals at the same time, but we only record their behavior periodically. So say you're going to follow these animals for 10 hours and you're going to collect a data point every 10 minutes of that 10 hours. And so you'd look at your watch, you'd wait for the beep, and at the 10 minutes, you'd look up, you'd take a snapshot in your brain of what every lemur was doing, and you'd write down what that lemur was doing at that scan. So female 1 was eating an unripe fruit, female 2 was resting, juvenile 1 was playing, male 1 was sleeping, and you'd repeat that every 10 minutes. And so you'd have a really nice record of what everyone in the group was doing on average, but you might lose some of the detailed information you could pick up from a focal follow. And so you really have to figure out what kind of method is the best way for you. And I don't want everyone to get lost in the details, but what I want you to take away from this is that scientists get to make a lot of choices in how we go about answering the questions we have. There's not necessarily one right way to do any particular project, and no single approach is perfect. There are always going to be things you miss from any method that you use, and sometimes it's going to be important to use more than one method, and that this is okay. We can't learn everything from a single project, but we are going to generate information to answer that question, even if it's not every piece of information. Okay, so let's say you make all of these decisions, you figure out how many animals, how many social groups, what time of day, what season, focal versus scan, and you figure out all those details and you collect a ton of data. And then you get home and now you have a lot of work to do to analyze those data. And so the first thing you need to do, which is usually really time consuming, is that you need to convert those handwritten notes into something on the computer. And this takes like a lot of time to type up these handwritten notes. It can take hours or days depending on how many data you collect. This is also usually when I realize all the mistakes I made in writing notes, like spelling animals' names wrong, or like, spelling species names wrong. So you get to correct a lot of those errors as well and really clean up your data. And once you've got things in a computer, you can start doing fancy analyses. And so I have some illustrative graphs here, which, disclaimer, are not based on real data. These are just for visualization. But say, for example, you collected all these data in the rainforest on these red-bellied lemurs and on these white-fronted brown lemurs, and you're able to generate these graphs of what the lemurs are eating. And so these graphs are called pie charts, and they represent the full diet of the animal. And each slice of the pie represents the portion of that diet that belongs to a particular food item. So in the red-bellied lemur case, you can see that most of their diet, the biggest slice of the pie, comes from fruits. And then they have sort of equal slices of the pie that are in flowers and leaves, and then other represents the smallest slice of the pie. And then we can compare what that pie looks like in red-bellied lemurs versus in these white-fronted brown lemurs. And what I hope you can see is that the slice of the pie that represents fruit is much bigger in the white-fronted brown lemurs, and the slice of the pie representing flowers is much smaller. So based on these graphs, we could infer that red-bellied lemurs eat more fruit and white-fronted lemurs eat, red-bellied lemurs eat more flowers and white-fronted lemurs eat more fruits. And again, to point out, these are not real data, so don't take this as what these lemurs are actually eating in the woods. And we might be interested in using these data to answer a different question, which is what are lemurs eating in different seasons? So maybe we find that in the rainy season, the animals are eating much more fruit, which makes more sense because there's more rainfall and maybe fruits are more abundant. But in the dry season, they're eating more leaves, maybe because those fruits aren't around all that much. And so you can see how we can use this kind of behavioral observation to generate a lot of information about what these animals are doing. And so this is really cool. But of course, there are also challenges to this method. And like I said, no method is perfect. And also some of these methods are easier than others for us to do. And the challenges of behavioral observation also include that it's really physically difficult at times. I personally tend to fall a lot in the forest. And on top of that physical challenge of just going up and down all day long, you might be spending a long time away from home. You might be spending weeks or months away from home. I have colleagues who spent years collecting behavioral data, and you're going to be living probably under pretty simple living conditions, eating, the same food over and over again. Maybe you don't have access to a good bathroom, and you're also going to be facing the elements. And I just love this picture of Marina's team from Sinjarivu, a different rainforest site, just like smiling through the most rainiest of the rainy season ever, and trying to collect data under those conditions is not easy at all. And on top of it being hard for us, there's also limitations of the data we're collecting. And that the biggest one is you can really only record what you see. And so a lot of the time the animals might be up in the trees, they're out of view, they're running around, you just don't know what they're doing at those times. And you might be missing things that they're eating. So you might have a lot of missing data. And maybe there's a whole day where you just have no idea where that group went. You never found them that day and you're missing an entire day. And then the other downside is that we can really only collect behavioral data on lemurs that allow us to collect data on them. And so many lemurs are really scared of people. And if you were to show up in their habitat, in their home, they would run away from you or they'd scream at you. And they wouldn't behave normally. And of course, we want to collect data when they're behaving normally. And so a lot of lemurs that are used for this kind of behavioral research or for ecotourism have been habituated to have people around. They're used to people being around them and they're not scared of them anymore. And so we can more easily collect data on them. But that means we're limited to our behavioral data to animals that are okay with us being around them, which means there's a lot of animals we just can't study. And so because of these limitations, we might want to turn to other methods than just behavioral data. And this might be where you're thinking, like, what happened to the poop? Because I thought we were going to do behavioral data and poop collection. And so maybe there's other types of methods that we can use to answer this question as well. And so now we're going to use some fecal methods. And so one obvious one we could do with feces is we could just visually inspect the poop for things that the animals are excreting that are evidence of the food they're eating. So these lemurs might be pooping out full seeds that we could then figure out what the fruit is. You might see bits of insect exoskeleton like wings come out in a lot of animal poop. And it could also be that there's undigested leaf matter in there that we could use to figure out if they're eating leaves. And so we're also going to be collecting feces from these animals. And so you go out in the rainforest and you stare up and you wait for animals to poop, which it turns out these animals poop a lot. And so this is what some of the poop of these red-bellied lemurs and white-fronted brown lemurs looks like. And as you can see, it's sort of chock-a-block full of seeds, actually. And it turns out these guys eat a lot of fruit and poop a lot of seeds. And if you're lucky, you can figure out the fruit that those seeds came from in the case of this pandanus. And sometimes you get really surprising seeds that are absolutely enormous. And so I know this picture doesn't do the scale justice. So I've got one of Marina holding one of these seeds. And so this is actually a seed that was basically pooped on our heads by a red-bellied lemur, which was kind of an amazing experience. And it was so big, it could have, maybe like given me like a little cut on my head. This thing was so big. And if you spend enough time watching these lemurs poop and you collect enough samples, you can actually find a lot of different seeds in their poop. And this is one of the ways that scientists realize that these animals are actually really important seed dispersers. And one of their major roles in the ecosystem is to eat fruit and poop out the seeds intact. They don't destroy the seeds, they poop them out intact with their own fertilizer starter kit, and they spread them out all over the rainforest. And that's from where we get a lot of new tree growth, is from these lemurs. And it was all through studying poop that scientists kind of figured this out. But of course, there are also limitations to this method. And so maybe you end up coming away from your field season finding 25 different types of seeds corresponding to 25 different types of fruits in lemur poop. That's really cool. You can say with real certainty that these animals are eating these seeds and pooping out this fruit. But this doesn't tell you anything about the foods that aren't seeds. We have no idea just from the feces if these animals were eating flowers, if they were eating leaves, if they were eating insects. We don't know. And we also can't be sure that we didn't miss any seeds. Maybe there are really, really tiny seeds that we just didn't see or we couldn't find. And so we can only say with certainty what we did see, but we can't say with certainty what we didn't see. And if you thought this was all hard enough to study in these brown lemurs, I want to now paint the picture of what happens when we try to study Marina's lemurs, which are these guys. So this is the mouse lemur that lives in Marajejy National Park. It's ridiculously adorable, but it's also nocturnal. It is really, really fast. It is really tiny. This guy weighs about 10 nickels put together, so it's super tiny. It forages alone, so not in social groups, and it can also hibernate for months during the year, so it might just be like missing from the forest because it's busy hibernating. And if you think studying this guy is bad enough, the reality of what you're looking at is this in the forest. And so you are trying to collect behavioral data on what this animal is eating all night long, and this becomes really, really challenging. And so there's a couple of tricks that you can do to help figure this out, which is the first, that collars are an absolute must. You have to collar these animals to be able to find them. And so Marina uses these live traps, which are really safe for the animal. It's a long metal sort of rectangle, and you put a nice piece of banana or mango in one end. The lemurs run all the way in and the door closes shut. And because the trap is so long, there's no way the animals can get caught in the door. And so this is a really safe method to trap these animals. And then we can bring them back and put a collar on them. So here's a mouse lemur that we've put a collar on. These are really, really tiny, lightweight collars, so they're not a burden to the animal. While the animal's in hand, you can check it for health. You can collect any measurements you might be interested, like the size of its head or the length of its limbs. And then we keep the animal at camp for the day to help it recover. We give it some banana, we give it plenty of water, and then they hang out with us in the shade during the day because these are nocturnal animals. So this is when they'd normally be sleeping. And then at night, back when they'd be waking up naturally, we can release them in the exact same spot we found them. So they're going about their night now, going about their foraging activity, and the difference is that they have this collar on them whereby we can identify them and follow them. So in addition to collaring, also using the scan method is a really good idea because these are nocturnal animals, and in order to see what they're doing, we need to use flashlights. And we definitely don't want to keep a flashlight on this animal continuously. That would be probably not great for the animal to go about doing its business and go about doing its evening. And so instead, maybe we're going to take a 10 or a 15-minute scan where every 15 minutes we're going to turn the flashlight on really quickly, see what it's doing, and turn the flashlight off immediately. And so this creates much less disturbance for the animal. And just be prepared that you're going to have a lot of missing data. A lot of these scans are going to be just, I have no idea what that animal was doing or where it was. And also be prepared that it is going to be impossible to get fecal samples from this animal unless you get them while it's in hand on one of these captures. And my summary for this section is, nocturnal work is not for the faint of heart, which is why I don't do it and why Marina does. So I tend to work on diurnal day-living lemurs because they're a lot easier to study. And Marina is really hardcore and goes for these nocturnal critters, and hats off to her. All right, so in the last section, I want to talk about the types of questions that can't be answered by observation alone. And these are questions we're now going to turn to science experiment for. And experiments are really, really useful for a couple of reasons, in that we have much greater control over the study. We can ask very specific questions, and we can be very confident about our results. And I hope that'll become clear as I walk through some of these examples. And the best place to do lemur experiments is at the Duke Lemur Center here in Durham, North Carolina. But what's really important to know is that we never harm our animals in any way doing any of our science. I think the word experiment can sound really scary to people, like we're going to do things that are really scary, but you can do a ton of experiments that are super safe and super insightful and super informative without doing anything scary at all. And so that's one of the experiments we're going to walk through today. And those are the types of experiments that we do at the Duke Lemur Center. And just like observational work, experimental work also begins by having a question. You have to have a question. And so for this particular experiment, we're going to ask this question of how do lemurs choose what water to drink? How do they know what to drink? And this was part of the dissertation work by my good friend and colleague, Dr. Caroline Amoroso. So this was part of her PhD work that led to her becoming Dr. Caroline. And Caroline in particular studied these guys. This is another type of brown lemur. So again, not very brown, but they're called brown lemur. And she studied these guys in Madagascar, but also these guys and other lemurs at the Duke Lemur Center where they all live. And so Caroline had this hypothesis or this idea that lemurs drink clean water to avoid getting parasites. They're purposefully choosing water that's really safe for them to drink so they don't get sick. And this makes total sense. But how do you test this in Madagascar? Because lemurs drink what water's around. So how do you know what they prefer? And that's why Caroline came to the Lemur Center to do this experiment. And so what Caroline did was she gave the lemurs a choice. Do the lemurs want to drink really good water, or do the lemurs kind of want to drink nasty water? And remember that this water isn't harmful because no research that we do harms the animals. And so what Caroline did was she gave lemurs these bowls of really fresh, great local Durham tap water. And then she gave the lemurs bowls that were contaminated with feces, but those feces were sterilized. So we killed off anything that could hurt the animals in those feces, but that poop still provided the signal to the lemurs, like being kind of murky or maybe being kind of smelly, that maybe they don't want to drink it, but it was totally safe for them if they wanted to drink it. So in experiment 1, Caroline gave them these two options, this clean water versus this water that was contaminated with sterile, not harmful feces. And she did this with a bunch of lemurs. And I've got a video here that I'm hoping I can get to play. Great. So you can see these ring-tailed lemurs out in the forest at the Lemur Center. They've been presented with these 2 bowls of water, and they're sniffing the sort of grosser looking but safe one, and they don't like it at all. They don't like that any one bit, and they go instead for the nice clean bowl of water. Exactly what she predicted. And then Caroline repeated this experiment again. And so she gave them the same clean tap water bowl. And then she gave them the contaminated bowl that was filtered. And so she removed a lot of the extra gunk from that to create a less strong signal, a weaker signal, to see if even at this weak signal, the lemurs could still detect the difference between what the clean water was and what the sterilized contaminated water was. And then finally, Caroline did something really weird, which is she added blue food coloring to the different conditions, the clean water and the contaminated water. And she had a very specific reason for doing this. It wasn't just because Caroline likes blue things like her shirt is. Caroline had this idea that maybe the lemurs are drinking clean water because that's all they've ever had, and that's what they're used to. And so the situation for us would be like, let's say every day of your life you've had orange juice in your house, and you drink orange juice because it's always been in your house. And one day your mom or caregiver comes home with apple juice and says, do you want orange juice or apple juice? And you've never had apple juice before. So you pick the orange juice because you're used to it, not because you prefer one or the other. You pick one just because that's what you're accustomed to. And so maybe these lemurs are drinking this clean water because that's what they're accustomed to. And so this blue water should throw a wrench in that because lemurs aren't used to drinking blue water. And so this was a way to test that. And so Caroline repeated these experiments across like 60 different lemurs, many different species. And what she found are these results, which is that across the experiments, if we look at the number of drinks, that's what's going on the height of this on the y-axis, that these lemurs really prefer to drink the clean water across all conditions. They're a little less good at discriminating between the sort of filtered trial versus the most extreme ones, but they're still really good at drinking this clean water and preferring to drink this clean water. And so using this experiment, Caroline was able to say with some confidence that at the Lemur Center, lemurs avoid drinking water that's contaminated with feces. And summing this all together across this whole talk, what you might not have realized that we're doing is we're actually tracing something that's called the scientific method. And so the scientific method is a method of acquiring knowledge in science that's been around since the 1600s, and it involves the observation and the experimentation to test hypotheses and predictions. and really just to answer questions. This is how we as scientists answer questions. And so the scientific method follows a consistent flow whereby you have a question, you collect evidence, you analyze that evidence, and you report your findings, and then you rinse and repeat ad nauseum. So every time you report your findings, that feeds back into the next question you have. And what's nice about the scientific method is that this is the method that's used by scientists all across the globe. We all have different questions, we all use different ways to answer those questions, but we're all following these steps. And so we can all be pretty confident about what every other scientist is finding and how they're finding that information. And so this leads to what's called reproducibility in science. And this is really important, that we're basing our scientific findings on evidence and not on things we just think or guess. It's always based on evidence. But what this also means is that lemur science and all science is for everyone. You don't need to be a fancy professor to do science. All you need to do is have a question and come up with an idea for how you're going to answer that question using evidence. And so I'm really hopeful that you, the audience, will spend maybe part of today or part of next week thinking about the questions that you would have of lemurs, the type of science you would want to do with lemurs, and how you would go about asking those questions. And this could be as simple as reading lots of information on the internet. This could be watching hours of YouTube footage of baby lemurs playing to see if there's any differences between this lemur or that lemur. It could be watching footage on YouTube of lemurs eating and seeing how they chew if they use different teeth. There's lots of things that you can do to do lemur science right in your own home that don't require going to Madagascar and don't require being a fancy professor. And so that is the end of Lemur Science 101. And I hope you guys will come back and join us next week as we progress to Lemur Science 201, where we take a lot of this background information on why lemurs are so cool and how we go about studying it and start applying this knowledge base to like real questions that Marina and I are asking in our careers. And we flipped a coin, and I'm gonna go first, and we're gonna answer one of my favorite science topics, which is how do lemurs survive by eating leaves? And so that's gonna be what's coming up next week. And with that, I am super happy to answer any and all questions that our audience has about lemur science, the how. And so I'll hand this back off to you, Jesse.
Thank you so much, Lydia. What, you know what, in our lives, if we find someone or something to love as much as you love lemurs, then that's a successful life. Like right there, that's the definition. That was fantastic.
Thank you.
If you want to come out of screen share so we can have a bit of a conversation, that would be great. Fabrice, I'm going to come to you with your class's question in just a second, but I love this one to start from Carolyn in Massachusetts, and she wants to know, how did you get into lemur science? Like, where did all the passion come from?
So I got into lemur science by serendipity, which is that I came to college in North Carolina and we happened to have lemurs here. And I've always thought that if Duke had like an otter center or a rhinoceros center, that I would be studying otters or rhinoceroses. I don't know what the plural of that is. And so I think a lot of my passion is that I'm just like an excited person in general. And so it's not like from a young age, I was like, I'm going to be a lemur scientist. It's just, I got exposed to lemurs and I fell in love with them. So I think that's really important to think about exposure and going out and trying a lot of different things when you're a student and just figuring out what really inspires you. And I had like this very unusual path where I started off trying to be a professional ballet dancer, and that fell apart. And I came to college a couple of years late and I had no idea what I wanted to study. And we had these lemurs here and I was like, what is that? And I just fell in love with them and I haven't looked back.
Yeah, fantastic. I love that your story also included this art component to it, because we get so many people that feel that scientists, you know, they love it from when they're a really little kid and they have this plan when they're 4 years old and they do it straight and they become a scientist. And so many of the people we talk to have ballet or visual art or drama or dance or farming or who knows what in their career. And then they just follow that passion and become something like a lemur scientist. And yeah, fantastic. Thanks, Lydia. All right, I'm going to go to Fabrice joining us in Philadelphia. He has a question from one of his students. So if you want to just unmute your mic, come on in. And
Thank you, Jesse. Thank you, Dr. Greene, for this presentation. That was very interesting. I'm sure I cannot see my students right now, but I'm sure that they've enjoyed it tremendously. I have a question from Chris, one of our students here at the French International School of Philadelphia. He wanted to know, how many species of lemurs do you find in the Marojejy National Park?
Yeah. So in Marojejy, I think it's like, what is it, like 11? I'm double checking with Marina. How many in Marojejy? It's like 11 species. So we've got the silky shafak, we've got 2 species of brown lemur, we've got eye, we've got a sportive lemur, we've got a woolly lemur, 2 dwarf lemurs, 1 mouse lemur.
And a partridge in a pear tree.
And a partridge in a pear tree.
Yeah.
So yeah, it's something like somewhere between like 10 and 12 or something like that, like a ridiculous richness of lemurs. And this gets back into the talk from last week about why there are so many lemurs and why there are so many in any one place, which is because they all sort of have different strategies for survival. So they're all doing different things in that forest. So in most forests that you go to in Madagascar, you're going to have multiple lemur species, anywhere from like 4 to 12. And that's because they're all doing different things in that system.
Great question, guys. All right, Akash joining us all the way in India, had a ton of questions on YouTube for the presentation, and he wants to know about torpor and hibernation. So you talk about survival strategies. These are really unique ones. And so can you explain a little bit about how lemurs go into those processes, how many lemurs do, hibernation, and anything you can share?
So I'm going to partially answer this, and I'm partially going to punt it because Marina is going to do an entire talk on hibernation in dwarf lemurs. So make sure you tune back in, not next week, but the week after next week to talk about her, to see her presentation on torpor. I think the meat of the question is we don't really know how these animals go into hibernation. And that's one of the most fascinating areas of hibernation research right now, is literally the mechanics by which they become metabolically depressed. But the why goes back into Madagascar. And that, if these guys are going down the path of being fruit eaters and there's only fruit around for part of the year, there might not be that much food for them to eat in the other part of the year. So a strategy to cope with that fluctuating resource is to eat a ton of it when it's there, store it as fat, put it in your freezer for later that you can thaw in the winter. And so instead of having a refrigerator and a freezer system, they just keep that in their tail as fat. And so that's what fuels them when there's not food around. But the actual mechanics of how lemurs reduce their heart rate, how they slow down their breathing, how their brain activity slows down, all of that is a lot of what's being investigated right now. And I'm sure Marina will have more information about that.
2 weeks, we can't wait. Okay, we'll be there. That's very exciting. All right, I'm going to go back for a live question in just a minute, but we've got a bunch of questions on YouTube all around the same theme, which is how do you become, what sort of degree do you need to become a lemur scientist? And you highlighted at the end of your presentation that there are lots of ways to get into lemurs. So if there's citizen science programs that you know of that people could just contribute to off the street, great. But to become you, can you explain a little bit about what you might need to do education-wise?
Yeah, so I did an undergraduate degree and I majored in anthropology. And so as part of my undergraduate coursework, I took classes in primatology and primate conservation and ecology. And then after that was done, I ended up doing a PhD in ecology. And so I studied ecology and I used lemurs as my study system in my ecology degree program. And I think what's probably the most important thing is that as early as you can in your sort of academic career in college is to try to volunteer or get an internship or do an independent study with a lab at your school or a researcher that you find whose work you find fascinating. Ask them how you can get involved in their work, because most labs absolutely have undergraduate students that either do independent projects or that volunteer on projects or that even have like work study jobs and get paid to be scientists in college. And so those are all things that I did starting as early as my freshman and sophomore year in college. And some labs even have high school students that work with them. So it's never too early to get involved in ongoing lemur science projects. And don't be afraid to email and contact scientists and conservationists, because we're always happy to hear from students who are interested in getting involved. And we're usually happy to help students get involved in any way that they can. So don't be shy.
That's a fantastic message. I think a lot of students, when I was younger, I was afraid and leery to reach out to scientists. Oh, these professional people. If you reach out to a scientist at any university, and those emails are publicly available on pretty much every university website, you will almost always get a response back with just as like extreme enthusiasm. People are really keen to share their passion and knowledge. So Try that out. It's a great way to learn a lot. All right, we got so many people from Connecticut coming in the last few minutes. I love it. So welcome into all of them. So you've got, I like this as a joint question before we go back live to Philadelphia, from a person in London in the United Kingdom and from a Connecticut person. We've got about COVID. So how is COVID affecting conservation efforts in Madagascar, which is a really difficult region in general, and can lemurs catch it from people?
So I'm going to answer the first question first, which is that I don't know of any documented cases of COVID emerging in lemurs, but there are cases of like pugs and tigers having COVID. So I think probably most mammals can get it. So if a pug can get it, there's a good chance that any primate can get it. With regard to the second question about what's going on with COVID in Madagascar is Yeah, it's pretty scary. From my colleagues that I'm hearing, Madagascar has gone many of the cities into lockdown as well, which is creating the same sorts of economic hardships that are being created elsewhere. But a big problem for Madagascar is going to be the lack of a tourism season this year, because the borders are closed. And so a lot of livelihoods now derive from sustainable ecotourism ventures. And so that's what I'm hearing from my colleagues there, is that that's going to be something that everyone's really concerned about. There are a lot of Malagasy and international NGOs that are working to offset those consequences. So definitely do some Googling on the internet and find organizations that have partnerships with Malagasy organizations or that are Malagasy led, and find out ways that you can help offset those economic hardships.
Yeah.
So.
I mean, it's not a positive story, but I'm glad we highlighted that message. We did a global biodiversity festival back in May, where one of the main charities that we used to support during that time was something called Planet Madagascar, which works in the northwest.
Yeah.
So a lot of great organizations that work with local groups. And actually, you mentioned this, that how integral it is to work with the local scientists, local communities. And can you speak to that just for a second, if you could, how important that is?
Yeah, I mean, there are like some really exceptional scientists, I mean, in every country on Earth, and that doesn't exclude Madagascar. So there's a lot of people working on the ground, boots on the ground, getting a lot of science done out of Malagasy higher academic institutions. And so as a researcher, I believe wholeheartedly in collaborative team-based science. And so some of my team members for some of my projects might be Malagasy scientists, especially when the science is being done in Madagascar. And I think teamwork in science always makes the dream work, because it's always good to have as many ideas and as many sort of perspectives floating around as possible. And the only way we're going to answer some of these questions and face some of these big challenges is by people with a lot of big ideas from a lot of different fields coming together and putting their minds together. So.
Nice. Teamwork makes the dream work is like the greatest slogan. That's got to be on the cover, right? Yeah. Awesome. Thanks, Lydia.
These are good questions.
These are good. Seriously, every time, whether it's kids or adults, like we just get really in-depth questions. So I really appreciate that for everyone tuning in on YouTube. I'm going to go back to Philadelphia Live to take probably our second last question here. So I know you have one from Haviland, which is really exciting. You want to share that?
That's it. One from Haviland, a 7th grader at our school. She wanted to know, how do lemurs react to being taken away from their natural environment, and how would they handle the noise and the lights and, you know, the busyness of our own natural environment?
Yeah, so I would say for the captures that I've been on in Madagascar, they're usually really quick. So we would never maintain an animal for any longer than a day. And for most of that time, the animals are sleeping in like a bag that's hung from a tree, which is very similar to their natural environment. So the actual time that we spend in contact with them is very, very short, maximally maybe 20 minutes. So it's a really quick experience for them. And so I personally don't think that it's that scary, and it's not prolonged at all. And of course, the lemurs at the Lemur Center that we have under human care have been around people their whole lives, and they're totally used to us walking around them, and you know, they're not scared of us at all. So yeah, we try to keep the procedures as quick as possible, just to avoid disturbing the animals as much as possible.
One of the things I love the most in all these programs, whenever we do conservation, and whenever we talk about animals, is that there's such a slew of questions on the welfare of animals. Like everyone is always concerned with that above everything else. Like, how are the animals doing? I'm really glad you highlighted in your presentation that experiment and research need not mean harming the animals in any way. It can be fun things with blue food coloring and
yeah.
Dr. Greene, before we wrap up, you've highlighted so much. We're nearing the end of the broadcast. Is there any last message you want to share with kids about how they can get interested, involved, excited about lemurs the way that you are?
Just start reading and watching videos of lemurs, particularly if they're in sort of a natural setting. So avoid those, you know, pet lemur videos that aren't so great for the animals themselves, but learn up on your favorite species, be that a ring-tailed lemur, be that a shafak, be it a dwarf lemur. Get involved in learning about that specific animal and its ecology, the threats that face that animal, where it lives in Madagascar, and then find the scientists that are studying those animals and just ask how you can get involved. And I guarantee that those scientists are going to be really happy to talk to you.
Amazing. Thank you so much. One last recommendation before you guys go. So the Duke Lemur Center has been highlighted so much in this presentation. lemur.duke.edu is the website. There's so many amazing educational resources on the site, so check that out. Continue on with this series. I'm excited for Lemur Science 201. I feel like I'm getting a higher degree already. So come back with us next week. The week after that, we're so excited to hear from Marina as well on hibernation, on eating leaves, and more. Check out our past videos. You guys as an audience are so keen on lemurs that we just keep having to find more and more lemur scientists to fill the demand. So check out all our amazing lemur programs. Just type in lemur at Exploring by the Seat of Your Pants as YouTube. You'll find a lot of great stuff there. Dr. Greene, this has been so much fun. Thank you so much for joining us, and we really appreciate it.
Thanks for having me. This is fun for me too.
Awesome. Well, have a wonderful rest of your day. We look forward to seeing you next week. And for now, everyone,